Hashtag self-replicating RNA, the breakthrough that could explain life's origins. Hashtag Chapter 1, Hook, 0 to 10 seconds. Scientists have created a tiny RNA molecule that can copy itself. This sounds like science fiction, but it is real, and it happened in a laboratory in the United Kingdom. The discovery, published in the journal Science, could finally solve one of the biggest mysteries in all of science: how did life begin on Earth? For decades, researchers have debated whether RNA or proteins came first. Now, for the first time, we have direct evidence that RNA can self-replicate. This changes everything we thought we knew about the origin of life. Hashtag Chapter 2, The Discovery, 10 to 45 seconds. The breakthrough came from a team of researchers led by Dr. Johnny of the Medical Research Council Laboratory of Molecular Biology in Cambridge, England. They identified a remarkably small RNA molecule, named QD45, that can copy itself and its complementary strand. At just 45 nucleotides long, QD45 is the smallest RNA molecule ever shown to perform complete self-replication. Previous attempts to create self-replicating RNA had only managed to copy small fragments. QD45 can copy its entire sequence, making it the first true self-replicating RNA molecule ever created in a laboratory. The researchers published their findings in the journal Science on August 4, 2026. The paper, titled A Small Polymerase Ribozyme That Can Synthesize Itself and Its Complementary Strand, describes how QD45 can catalyze two key reactions required for self-replication. First, it can bind to a complementary RNA strand. Second, it can use that strand as a template to create a new copy of itself. This two-step process is the fundamental mechanism of biological replication, and QD45 demonstrates it using nothing but RNA. Hashtag Chapter 3, Why This Matters, 45 to 90 seconds. This discovery is groundbreaking because it supports the RNA world hypothesis, one of the leading theories about the origin of life. The RNA world hypothesis proposes that before DNA and proteins evolved, early life on Earth was based entirely on RNA molecules. RNA can both store genetic information, like DNA, and catalyze chemical reactions, like proteins. This dual capability makes RNA the perfect candidate for the first self-replicating molecule that gave rise to all life on Earth. For years, critics of the RNA world hypothesis argued that RNA molecules were too complex to have formed spontaneously on the early Earth. They pointed out that RNA requires precise chemical conditions and that the probability of a self-replicating RNA molecule forming by chance seemed impossibly low. The discovery of QD45 changes this debate dramatically. At only 45 nucleotides long, QD45 is small enough that it could plausibly have formed through prebiotic chemistry on the early Earth. The fact that such a small RNA molecule can achieve full self-replication makes the RNA world hypothesis much more credible. The researchers also demonstrated that QD45 can copy both itself and its template strand, closing the replication cycle. This means that QD45 can produce two copies of itself from a single template, a fundamental requirement for any self-replicating system. In biological terms, QD45 has achieved what scientists have been trying to demonstrate for over 30 years. Hashtag Chapter 4, The Science Behind the Breakthrough, 90 to 135 seconds. The QD45 molecule is a type of RNA enzyme called a ribozyme. Ribozymes are RNA molecules that can catalyze chemical reactions, just like protein enzymes do. The discovery of ribozymes in the 1980s was a Nobel Prize-winning achievement that helped launch the RNA world hypothesis. But until now, no ribozyme had been shown to fully replicate itself. The researchers used a technique called in vitro evolution to identify QD45. They started with a large pool of random RNA sequences and subjected them to repeated cycles of selection and amplification, favoring sequences that could catalyze RNA synthesis. Over many generations, the pool evolved to contain increasingly efficient self-replicating RNA molecules. QD45 emerged as the most successful candidate. What makes QD45 special is its efficiency. The molecule can copy itself in approximately 1 hour under laboratory conditions. While this is slow compared to biological replication, it is remarkably fast for a synthetic RNA molecule. The researchers also found that QD45 can tolerate mutations, meaning that it can evolve and improve over time. This is a critical feature for any system that is supposed to have given rise to life, because evolution requires variation and selection. The implications extend beyond the origin of life. The discovery of a small, self-replicating RNA molecule has practical applications in biotechnology and medicine. Self-replicating RNA could be used to create new types of diagnostics, therapeutics, and synthetic biology tools. Researchers are already exploring whether QD45-like molecules could be engineered to detect disease markers, produce therapeutic proteins inside cells, or serve as programmable catalysts for industrial chemical processes. Chapter 5, What comes next? 135 to 175 seconds. The research team has announced plans to take the next step toward creating a fully self-sustaining RNA replication system. Their goal is to combine QD45 with other RNA molecules that can perform complementary functions, such as producing the building blocks needed for replication or protecting the RNA from degradation. If successful, this would create an RNA-based system that can sustain itself without any external intervention, a key milestone in understanding how life could have emerged from non-living chemistry. The team also plans to investigate whether similar self-replicating RNA molecules exist in nature. Some researchers believe that remnants of ancient self-replicating RNA systems may still exist in modern cells, hidden within the vast complexity of contemporary biochemistry. Finding these remnants could provide direct evidence for the RNA world hypothesis and illuminate the path from chemistry to biology. Other scientists are cautiously optimistic about the findings. Dr. David Bartle, a prominent RNA researcher at MIT who was not involved in the study, called the work a significant advance that brings us closer to understanding how life could have started from simple chemical systems. However, he noted that QD45 still requires purified components and controlled laboratory conditions, which would not have been available on the early Earth. The next challenge, he said, is to demonstrate self-replication under more realistic prebiotic conditions. Chapter 6, Call to Action, 175 to 200 seconds. The discovery of self-replicating RNA is one of the most important scientific breakthroughs of 2026. It brings us closer than ever to understanding how life began on Earth and opens new possibilities for biotechnology and medicine. If you found this video interesting, share it with someone who loves science. Subscribe to TrendForge for more coverage of the latest discoveries in science and technology. And remember, the answer to one of humanity's oldest questions may be hiding in a tiny RNA molecule. Stay curious, stay informed, and never stop exploring.